Hello friends, in this module we will discuss the habitat and classification of archaea. Many archaeans are extremophiles. Some live at very high temperatures, often about 100 degrees, as found in hot springs, geysers and black smokers. Others are found in very cold habitats like glaciers in Arctic Ocean or in highly saline, acidic or alkaline water. However, archaeans are also mesophiles and have been found in environments like marshland, sewage and soil. Many methanogenic archaea are found in the digestive tracts of animals such as ruminants, termites and humans. Archaea are usually harmless to other organisms and none are known to cause disease. Recently, several studies have shown that archaea exist not only in mesophilic and thermophilic environments but are also present sometimes in high numbers at low temperatures as well. It is increasingly becoming recognized that methanogens are commonly present in low temperature environments such as cold sediments. Let us first discuss the classification of archaea. Archaea are classified into 5 phylums. The first is Crenarchaeota, the second Euryarchaeota, the third Korarchaeota, the fourth Thaumarchaeota and the fifth Nanoarchaeota. Let us first discuss Euryarchaeota. These microbes share many basic properties, but despite these commonalities, the Euryarchaeota exhibit a great deal of metabolic diversity and consists of 9 orders and 15 families. On the basis of habitat and catabolic activity, they can be divided neatly into 4 subgroups: Methanogens, Halophiles, Thermoacidophiles and Hyperthermophiles. Coming to methanogens, many members of the Euryarchaeota produce methane as an end product of their metabolism and are called methanogens. They are strictly anaerobic organisms and are killed when exposed to oxygen. This is due to the oxygen lability of a number of cofactors and enzymes present in the microorganisms. Let us now discuss the occurrence of these methanogens. Despite their oxygen sensitivity, they are found in a wide variety of habitats. The sediments of many bodies of water, especially anaerobic sediments of marshes, swamps and rice fields, serve as excellent habitats for these organisms. Areas that collect and process human waste such as moist landfills and anaerobic sewage digesters also have high numbers of methanogenic bacteria. In addition, methanogens are common residents of the digestive tracts of various types of animals including ruminants, the large intestine of humans, dogs and swine, and in the hindgut of cellulose utilizing insects like termites. The common thing among all these habitats is an anaerobic environment that contains carbon dioxide and a highly reduced substrate. Common substrates include hydrogen, several one-carbon compounds and acetates. When growing on hydrogen, methanogens convert carbon dioxide to methane and also release water, as can be seen in this equation. When methanol is used as the substrate, the following equation shows the reaction. Finally, Growth on acetate is also performed by a small subset of methanogens, as can be seen in this equation. Despite the limited number of methanogenic species that can grow on acetate, those that perform this metabolism are critical for carbon cycling in the environment. Methanogens have a significant impact on the Earth's atmosphere. Methanogens produce most of the methane in the Earth's atmosphere, and methane is a significant greenhouse gas and is a cause of global warming. Much of this methane production is associated with activities for which humans are at least partially responsible. This includes the flooding of rice paddies and the raising of domestic cattle, both of which create habitats for methanogens. Two examples of methanogens are Methanobacterium thermoautotrophicum and Methanococcus janacei. These are two of the first methanogens to have their genomes sequenced. The genome of Methanoautotrophicum contains 1.75 MB and 1873 proposed genes, while that of Methanococcus janacei contains 1.66 MB and 1729 proposed genes. Extreme halophiles are a diverse group of archaea that inhabit highly saline environments and are the most salt-tolerant organisms known. There are about 20 known species of extreme halophiles with diverse cell morphologies and cell structures, but they do share some things in common. They all require high salt concentrations to grow and are not viable in low salt medium. Extreme halophiles require high concentrations of salt, at least 1.5 molar, that is 8.8% NaCl, with an optimum between 2 to 4 molar NaCl and a maximum at 5.5 molar, that is 32% NaCl. Amazingly, saturated solutions of salt will still support the growth of some extreme halophiles. They are chemoorganotrophs using organic acids and amino acids as energy sources, and they require some growth factors. One of the first questions asked about these microbes was the nature of the molecular adaptations these organisms have undergone that allow them to survive in high salt environments. One of the main problems associated with high salt environments is that osmosis will pull most of the water out of the cell, literally dehydrating the cell if nothing is done to prevent it. Extreme halophiles accumulate inorganic ions inside the cell at concentrations that match the salt concentration outside the cell. A significant amount of information is known about the genus Halobacterium, and this information might apply to all extreme halophiles. In Halobacterium, a pump transports high amounts of potassium inside the cell such that the concentration of potassium is greater inside the cell than the sodium concentration outside the cell. The macromolecular constituents of the cytoplasm have been altered such that they still function in the presence of high potassium concentrations. With these adaptations, the extreme halophiles are able to live in environments where most other organisms die, and they therefore take advantage of a niche that is rich in nutrients. An example is Halobacterium salinarum, which is a rod-shaped archaea and was initially isolated from salted fish a century ago. Halobacterium salinarum first drew general attention with the 1971 discovery of bacteriorhodopsin, the light-driven proton pump isolated from the purple membrane of this organism. The resulting proton gradient is subsequently converted into chemical energy. The purple membrane of the halobacterium results in the entire saline water body turning purple like the Lake Hillier in Australia as shown in this picture. Let us move on to Thermoacidophiles. This branch of Euryarchaeota contains thermophilic and acidophilic organisms that thrive in the most acidic environments known to support life. Three genera are known: Thermoplasma, Picrophilus, and Ferroplasma. Let us first discuss Thermoplasma. These organisms thrive in acidic and high temperature environments. They are irregularly shaped and are spherical at lower temperatures. They are chemoheteroorganotrophs growing on organic compounds present in their environments and fastidious, requiring a number of growth factors to multiply. They can grow at pH values as low as 0.5. They may be motile by flagella. Two species of Thermoplasma have been isolated. Thermoplasma acidophyllum from coal refuse piles and Thermoplasma volcani from sites of volcanism. Their plasma membrane is strengthened by large quantities of diglycerol, tetraethers, lipopolysaccharides, and glycolipids. DNA is stabilized by nucleosomes. The genomes of both Thermoplasma acidophyllum and Thermoplasma volcani have been sequenced and have a reasonably high degree of sequence similarities. The genomes have sizes of only 1565 kb for Thermoplasma acidophyllum and 1585 kb for Thermoplasma volcani and are some of the smallest living organisms. Moving to Picrophilus. Picrophilus species are some of the most acid-tolerant organisms known. They were first isolated from samples taken from acidic hot springs and dry hot soil in Hokkaido. They are obligate anaerobes. They are also thermophilic and grow only between 40 degrees and 69 degrees. Their pH optimum is an astounding 0.7 and they are capable of growth at even pH 0.06. This is equivalent to 1.15 molar solution of hydrochloric acid, something capable of dissolving metal. Picrophilus species are irregularly shaped cocci with a diameter of 1 to 1.5 micrometer. They lack cell wall and have an S layer outside the plasma membrane. Moving to Ferroplasma. The first isolates of this genus were isolated from a bioreactor in Kazakhstan and an abandoned mine in the US. Ferroplasma cells are chemolithotrophic, oxidizing ferrous ions or pyrite for energy. Ferric ion is the end product of the oxidation. Hence, They are generally found in acid mine drainage, which are the outflow of acid from metal mines. The organism grows optimally at a pH of 1.2 and has a range of pH 0.1 to 2.5. They are pleomorphic, irregularly shaped cocci, as can be seen in the picture on your screen. Extreme thermophiles. The final branch of Euryarchaeota contains a group of thermophiles capable of growing at temperature up to 113 degrees. Many species from this group have been isolated at solfataras, which are the opening in the Earth's crust that emit sulfurous gases, and hydrothermal vents, so their temperature tolerance is not surprising. Their optimal growth temperature ranges from 75 degrees to 104 degrees. They are strict anaerobes and are motile biflagella. The two most common genera of this group are Pyrococcus and Thermococcus. Both of these organisms are capable of growth on peptides and carbohydrates, and these fermentative pathways depend upon tungsten containing enzymes. This is unusual because tungsten is seldom used in biological systems. It remains to be seen what the purpose of tungsten is in these enzymes and what advantage it gives to thermophiles. In contrast to many of the hyperthermophiles, these microbes grow without the reduction of elemental sulfur to hydrogen sulfide. This is important because hydrogen sulfide is poisonous and highly corrosive to stainless steel fermenters, the most common type in industrial settings. Organisms in these genera are of considerable interest as a source of thermostable enzymes. In the near future, a number of enzymes from these microbes may have industrial applications. Let us now discuss the next phylum, Crenarchaeota. The Crenarchaeota group is again composed of Thermoacidophiles and Hyperthermophiles, but these are phylogenetically distinct from physiologically similar Euryarchaeota. The Hyperthermophiles of this kingdom can grow at the highest temperatures ever recorded for life. They lack eukaryotic histones. Their DNA polymerase is unlike the Euryarchaeota, and they do not contain the cell division proteins MIND and FTSZ that are common to bacteria as well as eukaryotes. The best studied members of these groups are hyperthermophiles, and they are all found near volcanic habitats either on land or in the sea. The first genera is Sulfolobus. These grow in hot, sulfur-rich acidic springs. The optimum temperature is 70 to 80 degrees and optimum pH is 2 to 3. They are gram-negative with irregular lobate morphology. Sulfolobus is a strict aerobe and can grow chemolithotrophically by oxidizing hydrogen sulfide or elemental sulfur to sulfuric acid and using oxygen as the terminal electron acceptor. Given the low solubility of oxygen in hot water, Sulfolobus species may have systems to increase the oxygen uptake. Acidianus is another genus of Crenarchaeota found in hot springs and has a potentially interesting metabolism. It includes facultative anaerobes that use elemental sulfur under both aerobic and anaerobic conditions. This microbe is capable of using sulfur at either end of its catabolic processes, either as an electron donor or as a terminal electron acceptor. In the presence of oxygen, sulfur is oxidized to sulfuric acid and the electrons added to oxygen. Under anoxic conditions, hydrogen gas is reduced and sulfur is oxidized, with the two elements combining to form hydrogen sulfide. Acidianous cells are more or less spherical and not as irregular as those of sulfolobus. They can grow at temperatures from 60 degrees to 95 degrees and have an pH optimum of 2. Sulfolobus DNA has about 37% GC content and acidianous DNA has even lower, that is 31% GC content. This might be a bit surprising because high GC DNA is more thermostable than low GC DNA when melting curves are done in a test tube. However, under physiological salt conditions and in the presence of proteins that associate with DNA, even low GC percent DNA is stable at these growth temperatures. Pyrodictyum species have an optimum growth temperature of 105 degrees but are also notable because of their remarkable cell morphology. Irregular disc-shaped cells are bound together in a mycelium-like matrix that is attached to sulfur crystals, as you can see in the picture. The matrix is made of fibers that are hollow and consist of protein arranged in a helix-like fashion. These fibers bind cells together and also serve as anchors onto the sulfur crystals present. Pyrodictyum cells generate energy by oxidation of hydrogen using elemental sulfur as a terminal electron acceptor. Carbon can be obtained through carbon dioxide fixation or by the use of organic compounds. Pyrolobus fumari currently holds the record as the most thermophilic organism, having a temperature maximum for growth of 113 degrees. Its growth optimum is also the highest known at 106 degrees, and it will not grow below temperatures of 90 degrees. Pyrolobus cells are obligate chemoautolithotrophs. The electrons are coupled to the reduction of nitrates, sulfides, or oxygen, forming ammonium, hydrogen sulfide, and water respectively. The microbe is also extremely thermotolerant and is able to survive autoclaving at 121 degrees for over an hour. The next phylum is Korarchaeota. Direct isolation of 16S rRNA genes from an unusual hot spring in Yellowstone National Park revealed one organism that did not fit into any another phylum. This new sequence was placed into its own phylum, the Korarchaeota, and since that time, several other related species, including culturable isolates, have also been found. They are also known as Xenarchaeota. These isolates split very early in the phylogenetic tree of life and their properties may be similar to those of the earliest microbes on Earth. Research is just beginning on these organisms, and understanding their novel metabolic properties may give new insights into earliest life forms. Each of these 6 hot springs in Kamchatka was found to contain Korarchaeota. The next phylum is Thaumarchaeota. The Thaumarchaeota arises from the Greek thaumas, meaning wonder. This phylum was proposed in 2008 after the genome of C. symbiosum was sequenced and found to differ significantly from other members of Phylum Crenarchaeota. All organisms of this lineage thus far identified are chemolithoautotrophic ammonia oxidizers and may play important roles in biogeochemical cycles such as nitrogen cycle and the carbon cycle. It was proposed on the basis of phylogenetic data such as the sequences of these organisms' rRNA genes and the presence of a form of type 1 topoisomerase that was previously thought to be unique to the eukaryotes. Only one pure isolate of this group, Nitrosophila vienensis, from a marine environment has so far been obtained. Individual cells display an irregular cocoid shape. Let us now move on to the next phylum, Nanoarchaeota. In taxonomy, the Nanoarchaeota arises from Greek meaning old dwarf. Nanoarchaea, which were discovered in 2002, inhabit high temperature environments with an optimum growth of 90 degrees. They contain both the smallest known living cell and the smallest known genome of 480 kb. Members of this phyla have not been detected in pure cultures. This phylum has a single representative member named Nanoarchaeum equitans. This unusual archaea is an example This unusual archaea is an obligate symbiont of another archaea, Ignicoccus hospitalis. They grow and divide on the surface of Ignicoccus. Cells of Nanoarchaeum are about 0.4 micrometer in diameter and replicate only when attached to the surface of Ignicoccus. In the figure, you can see Nanoarchaeum equitans growing and dividing on the surface of Ignicoccus hospitalis. The hyperthermophilic archaea can grow at very high temperatures, the temperatures at which life seems to be impossible. The question is, how do their proteins and DNA remain stable at these high temperatures? Thermostable proteins from the hyperthermophiles are quite similar to that from mesophilic bacteria in their amino acid composition. What differs is the protein folding, which is responsible for their thermostability. Hyperthermophiles also produce the heat shock proteins, chaperones, which help in refolding of the partially denatured proteins. Over 80% of the total proteins can be chaperones in the case of hyperthermophiles, which is also responsible for the cell's overall heat resistance. If the stability of proteins at high temperatures is something to wonder at, the stability of DNA at high temperatures is even more surprising. Such a high temperature and the DNA does not melt apart. In some archaea, the stability of DNA is due to potassium diphosphoglycerate and reverse DNA gyrase. The potassium prevents the DNA from chemical damage and DNA gyrase brings about positive supercoiling in DNA which helps to stabilize it. Some archaea also have certain heat stable DNA binding proteins called SAT70. In Sulfolobus, this protein binds to DNA and increases its melting temperature by about 40 degrees. Coming to the applications of archaea in biotechnology, let us first discuss the importance of archaeal biomass in biotechnology. We have seen above that archaea are a diverse group which can survive in harsh conditions and thus have a lot more potential as compared to eubacteria. But their industrial application has not been exploited much except in waste treatment. Anaerobic sewage treatment of sludge is brought about by methanogens. A number of halophilic archaea hold great promise in degradation of organic pollutants and in the treatment of concentrated textile wastewaters containing a high salt load. Biosurfactant producing halophilic archaea can play a significant role in the remediation of oil polluted saline environments. Sulfate reducing archaea have been found to be able to degrade alkanes. A few sulfide oxidizing thermophilic archaea have gained attention because of their potential in the extraction of metals from sulfide ores. For example, Sulfolobus metallicus has been exploited for mineral biomining in a pilot scale bioreactor where it tolerated copper concentrations of 40 grams per liter. Let us discuss the archaeal enzymes of biotechnological interest. Apart from the potential uses of archaeal biomasses, many archaeal enzymes can also find industrial applications. The first example of such enzymes belongs to glycosyl hydrolase family required in starch processing industry where starch is converted into more valuable products like dextrins, glucose, fructose and trehalose. In all these conversions, high temperatures are required to liquefy starch and make it accessible to enzymatic attack. Currently, enzymes from mesophilic organisms are used, but they are likely to be replaced by thermostable archaeal enzymes. Some of these recently discovered enzymes include an alpha amylase produced extracellularly by the haloalkylphilic archaean Natrononococcus species and cyclomaltodextrin glucanotransferase produced by a newly isolated hyperthermophilic archaean Thermococcus species. The next example of industrially important archaeal enzymes are lipases and esterases from Archaeoglobus fulgidus and Sulfolobus shibitae, which have been purified and characterized and are of interest in detergent formulations and dairy industry. Hydrolases and isomerases from extremely halophilic archaea have potential applications in several biotransformations in the production of food supplements. One should not forget the thermostable DNA polymerases such as the PFU DNA polymerase from Pyrococcus furiosus, which is used in PCR. Archaeal metabolites for biotechnology. There are a few archaeal metabolites such as proteins, osmotically active substances also called compatible solutes, exopolysaccharides, and special lipids which may have potential industrial applications. Certain archaeans contain membrane-bound retinal pigments, bacteriorhodopsin and halorhodopsin, which enable them to use light energy to drive bioenergetic processes. Bacteriorhodopsin creates a proton gradient that can be used by an F-class ATPase to synthesize ATP in anaerobic environments. Bacteriorhodopsin, isolated from Halobacterium salinarum, is currently being commercialized by Cobell and Munich Innovative Biomaterials in Munich, Germany. Halophilic bacteria produce compatible solutes that maintain a positive water balance in the cell and are compatible with the cellular metabolism. These compatible solutes are excellent stabilizers for biomolecules. Archaeal lipids are an excellent source for the formation of liposomes with remarkable thermostability and tightness against solute leakage. Archaeal lipids have been proposed as monomers for bioelectronics for several reasons, the most important being that their bipolar tetraether structure offers novel opportunities for protein-lipid interactions which are of interest for the assembly of electronic devices based on redox proteins or enzymes. A new class of potentially useful antibiotics has been discovered in Archaea. A few of these archaeocins have been characterized, but hundreds more are believed to exist, especially within Haloarchaea and Sulfolopus. These compounds are important since they are different in structure to bacterial antibiotics and so they may have novel modes of action.